Hello, my name is Leila Porter, and I'm a professor of anthropology at Northern Illinois University. Today I'm going to be giving you a talk called How Many Pygmy Marmoset Species Are There? Assessing Biodiversity Using Non-Invasive Field Techniques and Museum Specimens. I want to start by making sure everyone is familiar with the term primates. So this is an order of mammals that includes the apes, monkeys, lemurs, lorises, and tarsiers. And all these animals share a set of common characteristics, including fingernails and toenails rather than claws, grasping hands and feet, large brains relative to their body size, good 3D vision, and small litter sizes. Now, the primates currently have 80 recognized genera and 709 species and subspecies, and that number keeps increasing. And that is because we are finding evidence that some of the animals that we thought belonged to one species are actually distinct. So, for example, in this year, 2021, we've identified a new species, the popalanger. And this animal had been observed before in the wild, but we gathered new information to indicate it was distinct enough from other langurs to give it its own species name. Now I'm going to review a little bit about some of the terms we use in taxonomy, and this is the study of the general principles of scientific classification. So I'll use the example of the chimpanzee. So this chimpanzee here obviously belongs to the order primates, and its genus is Pan, its subspecies is Troglodytes, and then we recognize subspecies of this animal, and so its full name is Pan Troglodytes troglodytes. So I'm going to review what we mean by these different terms, genus, species, and subspecies, quickly here at the beginning. So by a genus, we mean a class or kind of animals that have at least one common characteristic. So in the genus Pan, there's the common chimpanzees and the bonobo. Now, these two different kinds of apes both have nice broad front teeth and small jaws. This distinguishes them from other apes like the gorilla and the orangutan. On the other hand, these two types of apes also differ from each other. The bonobo is more slender, has less hair, and is less aggressive and characterized by female dominance, whereas the common chimpanzee is stockier and hairier and characterized by male dominance and much more aggression. Now, within the genus Pan, we'd have to figure out how to define species. So one common definition relies on interbreeding. So under the biological species concept, we say that if members of populations can actually or potentially interbreed, then they are the same species, regardless of their appearance. Now, it turns out that in the wild, chimpanzees and bonobos do not currently interbreed. There's a large river that divides populations. So in the wild, they do not interbreed, but in captivity, when they have been housed together, they can interbreed. So under the biological species concept, we'd have to say that these two animals are the same species, but that would undermine all the differences that we know exist in their physical appearance and their behavior, and also in their genes. So as a result, many people now rely on the phylogenetic species concept, and this defines species as members of a group who are descended from a common ancestor and who all possess a combination of certain defining traits. So in this sense, we're defining species as members who have a unique evolutionary history. So I have drawn a little phylogenetic tree here at the bottom. So this is a little evolutionary diagram showing how the apes and humans are related And so if the branches are small, they share a more recent common ancestor, and if the branches are longer, they have a more distant ancestor. And we can look for fossil record, genetic evidence, and other lines of evidence to actually put some numbers on how long ago the common ancestor existed. And so in the case of the chimps and the bonobos, we know that about 2 million years ago, they shared a common ancestor, but after that point, the two lineages split. So we'd say those are two nice species that have a 2 million year distinct evolutionary history. Now, subspecies would then be variants within a species. So once again, we'd have to identify some clear ways that they differed from each other, but we wouldn't raise those differences to the level of the species. So they're a little bit more minor, and presumably they shared a common ancestor more recently. And so the chimpanzee has 4 recognized subspecies, whereas the bonobo has just 1 species known. No subspecies are recognized. So how are we going to make these determinations about whether animals belong to the same subspecies or species? So if we use the biological species concept, we rely on breeding experiments. But this is often impossible to do for wild animals and also usually unethical. We could rely on physical appearance, for example, how hairy they are, how slender or stocky, or we could look at genes and we could compare genetic sequences of one animal to the next. And this is what has been done more frequently over the last couple of decades. Now, I want to introduce just one last term, and that is a clade. And this is a group of biological taxa, such as species, that includes all descendants of one common ancestor. So I've taken that little evolutionary tree of the apes and tipped it on its side. And I've also focused just on the genus Pan. So we could show that Pan paniscus, the bonobo, and all the common chimpanzees shared an ancestor approximately 1 million years ago. And then I've oversimplified this a bit. We'd say that the common chimpanzees, the Pan troglodytes, all had a common ancestor at 500,000 years ago, and then split into these 4 different subspecies since then. So we could say that the clade would be all of these types of Pan troglodytes, so these subspecies with their common ancestor 500,000 years ago. But we could also look at the clade further back and say that all the chimpanzees and bonobo share a common ancestor 1 million years ago. So this is what we mean by a clade. Now, we could map out where the subspecies of chimpanzees are in Africa, and you'll note that they are separated geographically. So often there are rivers that divide populations that are genetically distinct and have their own evolutionary history. Now, it turns out that some animals look very similar across a broad geographic range, but when we collect DNA from those animals from different locations, they appear to be genetically quite distinct. And so these are termed cryptic species because they're hiding. So hiding in plain sight because they look the same, even though they are distinct. So as an example, within the order primates, the mouse lemurs are small nocturnal lemurs that live on the island of Madagascar. And they're found across the island, so on north, south, east, west, and everything in between. And we used to call them all one species, Microcebus murinus. But after people started looking at the DNA of animals from different populations, they realized they were quite distinct, and they are now recognized as 24 different species of mouse lemurs. So these were all cryptic species waiting to be discovered through DNA testing. So now that we've laid out the basic background, I want to turn now to my question, and that is, how many cryptic species are there in the world? And I was specifically interested in one kind of monkey in South America, and the answer to that question lies in getting DNA samples. Now, to do that, we obviously have to get genetic material from wild animals. So in the old days, so a couple decades ago, we would have had to have trapped an animal. And this is a rodent in a little trap. And unfortunately, this process is quite stressful for the animal and can result in injury. Once the animal is trapped, we'd have to grab it and take blood or tissues and hair, and those could be used for genetic analyses. So fortunately, we have new methods, and the two I'll talk about today are fecal samples and ancient DNA. Now, the nice thing about trying to collect fecal samples is that monkeys and apes have to poop frequently throughout the day. So if you are able to follow a monkey or an ape, you should be able to see where it poops. Then you can try to look for that sample down below if the animal was up in a tree. Now sometimes this is a little difficult because the poop actually ricochets off leaves and may get buried in the leaves on the forest floor. But if you practice and you've got good eyesight, you probably can collect some good fecal samples. Now, if you're unable to do that, you can recruit the help of dogs. So there are some conservation teams that work with trained dogs, and what they do is they train those dogs to look for the scent of a particular animal and the scent of their poop. And so this is a group pictured here called Working Dogs for Conservation. Those dogs were trained to find the poop of one subspecies of gorilla in Cameroon. Now these gorillas are very shy and rare and live in very mountainous terrain, so it's difficult for people to follow them. On the other hand, dogs can be trained to look for gorilla dung, which is actually a large pile. And so the dogs can find gorilla dung, and they did so to try to help collect poop samples that could then be used for genetic analyses. So if you find a fecal sample and you collect it, you can bring it back to your lab and you can extract the DNA from it. Now obviously you're going to have to eliminate all the DNA that came from whatever that animal ate recently. So that could be plant matter, insect matter, or if the animal is carnivorous, the other animals it ate. So you have to remove all of that extraneous DNA and just focus on the DNA of the animal that pooped. And the reason that there is DNA in the poop sample is that cells from the intestines get sloughed off during the process of digestion, and so those intestinal cells have the DNA of the animal in them. Now, the other technique that we can turn to is the extraction of ancient DNA. So the phrase ancient DNA refers to being able to extract DNA from degraded material. Now, the most famous example probably of ancient DNA being extracted is that of the Neanderthals. So the Neanderthals were a type of modern human that lived about 20,000 years ago and later, as in older. And so the remains of those Neanderthals have been fossilized. but not all the original organic material of the Neanderthals' bones have turned into the fossils, so they have not been fully mineralized, and so those little degraded segments of the Neanderthals' original DNA from inside the cells of that bone can be extracted. And in 2010, a research team in Germany at the Max Planck Institute for Evolutionary Anthropology managed to extract enough DNA to sequence the entire Neanderthal genome. And this has given us great insight into the Neanderthals, as we can compare their genome to modern humans. And in fact, we know that there was interbreeding between Neanderthals and modern people because of these analyses of this ancient DNA. Now, the other place that you can get ancient DNA is from natural history museums. So in the 1800s through the 1950s, scientists went around the world exploring and collecting animals. Now, they would shoot those animals and then bring them back to a museum where they were preserved. So usually there is a skeleton from the original animal, the fur or pelt, and sometimes these were mounted for public display in the museum. But if more than one individual of a given species were collected, then they put those in storage. And so some of the big natural history museums actually have very extensive collections of primates. And it turns out that in cleaning off the skeletal material, in some cases, not all the original flesh was removed. And so that flesh is still on the skeleton. And this little dried material is referred to as crusties. So here I am holding up the skull of a little primate, and I can look in and find this little pieces of dried flesh. And I can remove it without damaging the skull, with the permission of the museum, of course. And we can use that crusty material to extract DNA. Now, in 2013, one research group looked through museum collections to try to collect crusties of guenons. These are a type of African monkey, and they're a very diverse group. And so it was a little unclear how many species were actually present and what the evolutionary relationships were among them. And so with the help of these crusties collected from museum specimens, these researchers were able to better map out the relationships among the living guenons. So I have been interested in a different group of monkeys, the calatrichins. These are monkeys that live in Central and South America. They're small little monkeys that you might have seen at the Brookfield Zoo and Lincoln Zoo and other zoos. They include the calamicos, the tamarins, the lion tamarins, and the marmosets. So currently we know of 7 genera, and the littlest of these monkeys is 100 grams, and the largest is about 600. Now, although I said that all primates have flat fingernails, this group of monkeys have actually re-evolved claw-like nails that allow them to cling to trees, and that allows them to forage from vertical trunks. And so I'm going to be talking about one little monkey, the pygmy marmoset, that clings to trunks and gouges holes in those trees to cause the tree to produce exudates. So some of the calatrichins feed on trunk exudates, and others search for insects on tree trunks. Now, these monkeys live in a diverse set of habitats, but the majority live in the Amazon basin. So here's a picture of the pygmy marmoset, and they belong to the genus Subwella. So they are the smallest of all the callitrichins. They would be only about this big, and then their little tail coming down. They are the most specialized for exudate feeding, and in this picture you can see an individual clinging to a tree trunk, and they have special incisors that are sharp that allow them to gouge holes in the tree, and then they can come back after the tree has produced the exudate to seal that wound and eat the exudates. So they're harvesting their own food from the holes that they've made. They're most common in riverine forests, and that's because the trees that they like to gouge are most common along rivers. So I was quite interested in the pygmy marmosets, and I became convinced that probably the diversity of the pygmy marmosets was not well known. So I wanted to find out how many pygmy marmoset species there really are. So when I started this research question, this was the known distribution of the pygmy marmosets. So you can see the map of South America there, and the pygmy marmosets are found in the red area. And I focused in the middle map just on Peru, and you can see that one subspecies of pygmy marmoset, shown shaded in green, was in the north of Peru, and then another subspecies in the south of Peru. And on the other side you can see drawings of these two subspecies, and the thought was that you could tell them apart by differences in their fur coloring. Now, in 2011, a German primatologist, Tauschek, surveyed the tamarins in Peru, and he looked at DNA samples of different populations from the north to the south, and these animals had been considered subspecies based on differences in their pelage. So some have different eyebrows, slightly different colors on their arms and backs. But his genetic analyses revealed that these populations were genetically distinct and at the level that warranted them to be called species. So 8 different species of tamarins are now known in the country of Peru. Now, if we go back to our pygmy marmosets and overlay their distribution onto the tamarins, you can see at the top there with the diagonal lines, those are supposed to be the northern subspecies of pygmy marmoset, and then below that with the grid lines, that was supposed to be where the southern subspecies of pygmy marmoset lived. But it seemed very odd to me that there would be only 2 subspecies of pygmy marmosets in Peru in the same region where 8 different species of tamarins existed. Now, there could be some reasons why one group would be more speciose than the other, but it also occurred to me that what we were looking at was a case of cryptic species. So the pygmy marmosets appear similar in Peru, but are they really? And so that's the question I wanted to address. So how to go about addressing this question? First, I knew that we could collect fecal samples. Then from those fecal samples, we could extract DNA. Second, I knew that we could use museum specimens to retrieve crusties and do ancient DNA extraction. And then from those same museum collections, we could take pictures of the animal's fur or pelage, and I wanted to measure skulls to see if they differed in their morphology. So there's the shape of their skulls. So I want to acknowledge my co-authors on this project. So first, Dr. Stella de la Torre is at the University of San Francisco in Quito, and this is in Ecuador, and she knows everything about pygmy marmoset behavior. She is definitely the world's expert in pygmy marmosets, and she's been observing them and collecting data on them at 2 sites in Ecuador for a couple of decades. Another colleague, Pedro Perez Peña, is at the Institute for Investigation of the Amazonia in Peru, and he has looked at pygmy marmosets at 1 site in the Peruvian Amazon. And then my colleague, Dr. Liliana Cortés-Ortiz, is at the University of Michigan, and she has a genetics laboratory where she has extracted DNA from feces and crusties of other kinds of primates. So what we did, this research team, is we collected fecal samples at 2 sites in Ecuador, and then we took photographs of animals in the field in Ecuador and Peru. And then I visited museum collections here in the US. So I visited the Field Museum in Chicago, the Museum of Zoology at the University of Michigan in Ann Arbor, and then the American Museum of Natural History in New York City. Now I photographed the pelage of those animals in the museum collections. So here you can see the back, underbelly, and front of one pygmy marmoset that is in one of those museum collections. I collected crusties from inside the skull, and then I measured the skulls. So previous research had been done on tamarin skulls. So a tamarin skull is pictured there, labeled A, and the diagram shows different landmarks on a skull. So these are usually where different bones have come together, and there's a suture. So this is where 2 bones come together, and you can see that you can measure a skull many different ways. Now the pygmy marmoset skull pictured with the letter B is similar to the tamarin, so I was able to use these same landmarks for my measurements. So just as an example, the measurement NABR would be from the top of the nose to the middle top of the skull. So altogether, we had specimens from 14 sites. 3 contemporary field sites, 2 in Ecuador and 1 in Peru, and then 11 museum collection sites. So these were sites where researchers in the early 1900s through the 1950s had gone and shot and collected wild animal skeletons and brought them back to museums. This map shows where each of those locations is in Ecuador and Peru, and you can see that location 1 is right on the border of Brazil, Colombia, and Ecuador. Now, also, please note on this map that the diagonal lines are where that subspecies was previously recognized to exist, and the little half circles are where the second subspecies were thought to have existed. So first, let me show you the results of our mitochondrial DNA comparisons. So we looked only at certain segments of mitochondrial DNA that we extracted from the feces and the crusties. Now we had 26 museum specimens, so those crusties. We had fecal samples from 7 wild groups, and then we had other sequences from GenBank. Now GenBank is an online depository where researchers can upload genetic data. So there were 7 previously established genetic sequences for pygmy marmosets already on the web. Now, if you look at this little list of codes here, UMMC is University of Michigan Museum of Zoology, and then the number would be the specimen number of that museum animal. And FMNH is Field Museum of Natural History, AMNH is American Museum of Natural History, EC are those Ecuadorian field sites, and GB are the GenBank sequences. So this shows you a little cladogram, right? So if the little branches are very short, that means that the 2 specimens were very close together in terms of their genetic sequences in these mitochondrial DNA locations that we examined. And if the branches are very long, then they are farther apart or more distantly related. And at the bottom, you can see that we used an outgroup, which was other callitrichins, to establish more distant relationships. So these are large-bodied marmosets of the genus Calothrix. So not surprisingly, the other callitrichins and the pygmy marmosets are very different in the mitochondrial DNA sequences. But then if you look within our pygmy marmoset samples, one set of samples cluster in one clade, and you'll see on the map I show you, these animals all came from the northwest of our study locations. And there was a second clade 2, that all came from animals in the south of the range. And then I'll further show you that one small set of animals there down at the bottom, shown in dark gray, form a subset of that clade 2 south group. So we have 2 clades with one clade having a subgroup within it. So if we plot these on our map, the light gray, shown here, is where we see the clade 1 animals coming from. So it actually forms an arc, so forming sort of a letter C from Colombia west into Ecuador and then down into Peru. And then our clade 2 animals come from sort of the middle of this zone in Peru and Ecuador and then into Brazil, with the Peruvian and Ecuadorian samples forming this subgroup of the clade 2 division. So then we can look at the pelage. So I took all these photographs of museum specimens, and a previous researcher named Garbino and his colleagues had come up with categories for how to classify them. So you can see that they vary from having a reddish underside, that's what we call pelage pattern 0. Then there's pelage pattern 1 with a thin white line down the middle. Pelage pattern 3 has this nice white understory, and then we added pelage pattern 4, which has sort of this grayish yellow underside. Now in our study, we found none that fit in Garbino's category 2, so that was not present in the Ecuadorian and Peruvian samples. I mentioned we also had photographs from the wild, so up in squares labeled A and B, these are animals at the Tiputini Biodiversity Station in Ecuador. And although it's a little hard to see, the animal in figure A is eating some of these exudates. So it's pulling out the little sticky substance from a hole. But the important thing is you can see it has a nice little white belly. In contrast, animal B from that same place in Ecuador has a brown belly. C is a picture from another site in Ecuador, again a brown belly, and D comes from a site in Peru, a nice white-bellied little marmoset. So we could label are photographs based on the same pelage type categories that we used for our museum specimens. In terms of cranial morphology, in total I had 18 skulls that I could measure. Some unfortunately were broken, so that meant I had fewer to measure than actually are in the museums. Now, what was interesting is that our two different molecular groups, this clade 1 and clade 2, differed in the shape of their skulls. And where the measurements differed is in the side of their head here along their cheekbone. So we have zygomatic length being longer in one clade than the other, and mid-cranial height taller in clade 2 than the other clade. So there are differences in the morphology of the skull that match our two distinct genetic clades. So we then took all of our photographs of our museum skins and our wild animals, and we plotted them onto our genetic map that shows the clades. And we could then see, do the skins of these animals match the genetic types like we found with the skull? And the answer is no. So I mentioned that at the Tiputini site, shown here at the top, there's two animals from the same population, the same clade. So this would be clade of the population is 2, shown here in the dark gray. You have animals with brown bellies and white bellies in the same spot, and their genetic material does not differ. And so we did not see a nice correspondence between pelage type and genetic type. So in sum, our data indicate that we actually have two distinct species. These are not subspecies, just slight variations of each other, but two distinct lineages with their own unique evolutionary history that has led to genetic changes in their mitochondrial DNA and differences in their skull shape. But we can't distinguish them by pelage, so this definitely does make them seem like a cryptic species, animals that appear similar but are actually quite genetically distinct. Now, we did have sort of some holes where we need to go back and find material from some more locations to complete our map. And one of the places that I am working on is pygmy marmoset populations in Bolivia. So these would be the most southern marmosets. So I hope to have the genetic sequences of those animals available soon to see how they compare with the species further north. And fortunately, we've been able to team up with a European group of researchers to come up with an age estimate of how long the two distinct pygmy marmosets have been separated from each other. And the preliminary answer, hopefully we'll get this paper out soon, is about 3 million years. So these really do seem like really nice phylogenetic species. Now you might be thinking to yourself, what's the importance of documenting species? So you probably know that many types of primates are in danger of extinction. And this is due to a host of factors, one of the primary ones being habitat loss. As more and more people are born and expand into pristine habitats, the primates that used to live there lose their space. So this can be due to urbanization, to agriculture, to logging, to mining, to ranching. Another factor is disease. So increasingly primates are suffering from disease outbreaks. Some of these diseases they're actually getting from humans as we increase contact with these wild animals. So just as an example, there have been recent outbreaks of yellow fever in primates in South America, in countries like Brazil and Argentina. And of course, humans hunt primates in many countries around the world. And so all of these factors mean that there are fewer animals and they have fewer spaces to live in. So it's important that we develop protection plans for those different primates. And to come up with a good protection plan, you need to know what is the geographic range of a given species and what's its population size. And the reason for this is quite simple. If you think about it, if you think you have one species that lives over a wide geographic area, first of all, you might consider it to be at relatively low risk of extinction, but also it might be under different pressures in different regions, but overall you'd come up with one plan for that whole region. On the other hand, if you knew it was two distinct species and you need to protect each one, then each of those species might face different pressures in each of its areas. And so you're going to come up with different protection plans for each of those individual species in their smaller geographic range. So the International Union for the Conservation of Nature has a website called the Red List of Endangered Species, and it's very easy to use. You can put in any species you want and see what its level of threat is of extinction. And so fortunately, thanks to our work and the work of others in Brazil, we now have 2 species recognized. So we have our 2 nice genetically distinct clades that we can say are species, and their threat level has been elevated to threatened. So we now know it's not just one species over all of the Amazon, but rather at least 2. And so now people are planning accordingly. Now, some biologists argue that we're naming too many variants as their own species, and they call this taxonomic inflation. However, the alternative is just to call them all one species, and then I fear that we are not doing enough to protect those distinct populations from extinction. So I would argue that it is better to actually name each of those genetic variants than to lump them all into one species. So let me give you the example of a recent publication that showed that one type of butterfly in California was considered to be a variant of others. However, when scientists recently took the DNA from a museum specimen, so that ancient DNA, and compared it to the DNA of other California blue butterflies, they realized that it was actually a distinct species and it had already gone extinct. So it was too late to save that unique type of butterfly. So we want to avoid having this happen in the future by doing more work to properly document biological diversity. So the next species on my list is this monkey here. This one is Calamico geldii. Like the pygmy marmoset, it has been identified for many years as just one species. And I am quite certain that if we can figure out how to get fecal samples and ancient DNA of Calamico specimens, we will learn that it is actually more than one species. So with that, I would just like to thank all of these individuals and institutions, particularly the Jurica-Suchy Nature Museum and Benedictine University for inviting me to give this talk, to my colleagues who I mentioned earlier, and to the Chicago Board of Trade and Chicago Zoological Society for granting us funds for working on this project. And then, of course, to the 3 museums that granted me permission to work in their collections, and to the Ministry of the Environment in Ecuador for allowing us to collect the fecal samples. Now, if you'd like more details on our research project, I've provided the full reference here. This just came out this year in the American Journal of Physical Anthropology. So thank you very much for your time.